Okay, hi everybody. I'm Mr. J. I teach two of the students here, but I worked with HK Maker Lab last year, and they brought me in this year to give you kind of a very brief overview of biology. So I'm going to talk about this a lot today, but in general, I'm just going to kind of give you that baseline level so that no matter what lab you're in, you kind of are like, oh, I've heard that word before. I have some familiarity. I'm not going to teach you all of biology because we only have a few hours today, but I am going to give you some familiarity. And this packet that I made for you, you actually may have teachers who use worksheets similar to this. You may have actually done these worksheets before, which is fine. But I kind of, as much as I could, try to put together everything that seems like this is the base level you would need for biology. And you're going to take this packet home. You can kind of use it for your own research. I'm not going to use the word quiz, but we do have a check for understanding that I will give you. And basically, after every section, I'll give you this, and I'll say, do these questions. And then we'll grade it, and we'll be able to say, OK, how are you doing on this topic? Don't be scared. I don't want you to think, oh, OK, if I fail this, I'm out of Bioforce. You won't be. But it will tell you, oh, I really need to review gene expression, or I really need to review microscopy, right? So we're going to kind of go over those basic things. But the number one thing I'm going to say is, I am not going to teach you everything that you need to know. You're going to have to do some independent research once you find out your lab. So if you're in a lab that is dealing with like bones, then you're gonna really need to do some research on bones, right? And I'll give you my email. I'll write it up here, and you're welcome to email me, and I can say, hey, here's what I recommend you kind of review, some keywords. But you're all you all know how to use the internet, I'm assuming, and you know like the keywords to look up. But sometimes you just kind of have to have that base level knowledge. So I'm going to try to make this as engaging as possible. We're going to have a little break for lunch, and then we'll keep going through the afternoon. But if you have learned some of this before, I'll try to go pretty quickly so we don't get too bored. But if this is going too hard, feel free to slow me down, and I can stay with you. But the number one thing is I have this very intimidating 12-page check for understanding packet. But by the end, hopefully, you're going to be able to understand most of this. So we can get started. Are any of you currently in AP Bio? Are you okay? So this is maybe a good review for you. For the test? But some of this may be kind of base level review. But we're all by the end going to have this nice little floor in terms of our level of knowledge. All right. So let's get started. So part one today, I'm going to talk about the history of cells and introduction to cells. Part two and part three we're going to do probably after lunch, where we're going to talk a little bit more about genetics. Okay? So this is base level. So we're going to jump right into this. So the first thing we're going to talk about is the cell theory, which is just basically understanding what cells are. Some things I'm going to have you do with this worksheet, but some things it's a good idea to take notes. I see most people have notebooks. And if you don't have notebooks, maybe ask a partner for some loose leaf. But it's just a good idea to kind of get some notes down for these things. So I'm going to show you a quick video. We're going to try to make this as interactive and different media as possible. Just about the introduction to cells. And then what I'm going to do is I'm going to give you a new sheet, and I'm going to ask you to do a check for understanding question on it. This one should be easy, OK? But this is basically a nice little history of what we know about cells.
One of the great things about science is that when scientists make a discovery, it's not always in a prescribed manner, as in only in a laboratory under strict settings with white lab coats and all sorts of neat science gizmos that go beep. In reality, the events and people involved in some of the major scientific discoveries are as weird and varied as they get. My case in point, the weird history of the cell theory. There are three parts to the cell theory. 1, all organisms are composed of one or more cells. 2, the cell is the basic unit of structure, organization, and organisms. And 3, all cells come from pre-existing cells. To be honest.
Do you want me to go over that real quick? Oh, you need one more? OK. But hopefully you kind of fundamentally understand those three things. It's not necessarily that you need to have those all written down, but that you understand everything comes from cells. Cells don't just pop up out of nowhere. And then it's going to go into how we know what we know about cells.
This all sounds incredibly boring until you dig a little deeper into how the world of microscopic organisms and this theory came to be. It all started in the early 1600s in the Netherlands, where a spectacle maker named Zacharias Janssen is said to have come up with the first compound microscope, along with the first telescope. Both claims are often disputed, as apparently he wasn't the only bored guy with a ton of glass lenses to play with at the time. Despite this, the microscope soon became a hot item that every naturalist or scientist at the time wanted to play with, making it much like the iPad of its day. One such person was a fellow Dutchman by the name of Anton van Leeuwenhoek, who heard about these microscope doohickeys, and instead of going out and buying one, he decided to make his own. And it was a strange little contraption indeed, as it looked more like a tiny paddle the size of a sunglass lens.
Has anyone ever heard of Anton van Leeuwenhoek before? Yeah, he's very famous and is often kind of credited as the person who discovered the microscope. So remember his name, okay?
If he had stuck two together, it probably would have made a wicked set of sunglasses that you couldn't see much out of. Anywho, once Leeuwenhoek had his microscope ready, he went to town looking at anything and everything he could with them, including the gunk on his teeth. Yes, you heard right. He actually discovered bacteria by looking at dental scrapings, which, when you keep in mind that people didn't brush their teeth much, if at all, back then, he must have had a lovely bunch of bacteria to look at. When he wrote about his discovery, he didn't call them bacteria as we know them today, but he called them animalcules because they looked like little animals to him. While Leeuwenhoek was staring at his teeth gunk, he was also sending letters to a scientific colleague in England by the name of Robert Hooke. Hooke was a guy who really loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day. Something he brought upon himself as he made the mistake of walking in with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? Well, after Newton published a groundbreaking book on how planets move due to gravity, Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered, thankfully, after Newton's time.
You can learn something in
but
5
not
minutes.
his portrait, as sadly no one knows what Robert Hooke looked like. Fast forward to the 1800s, where two German scientists discovered something that today we might find rather obvious, but helped tie together what we now know as the cell theory. The first scientist was Matthias Schleiden, a botanist who liked to study plants under a microscope. From his years of studying different plant species, it finally dawned on him that every single plant he had looked at were all made of cells. At the same time, on the other end of Germany was Theodor Schwann, a scientist who not only studied slides of animal cells under the microscope and got a special type of nerve cell named after him, but also invented rebreathers for firefighters and had a kickin' pair of sideburns. After studying animal cells for a while, he too came to the conclusion that all animals were made of cells. Immediately, he reached out via snail mail, as Twitter had yet to be invented, to other scientists working in the same field, and it was Schleiden who got back to him, and the two started working on the beginnings of the cell theory. A bone of contention arose between them, as for the last part of the cell theory, that cells come from pre-existing cells, Schleiden didn't exactly subscribe to that thought, as he swore cells came from free cell formation, where they just kind of spontaneously crystallized into existence. That's when another scientist, named Rudolf Virchow, stepped in with research showing that cells did come from other cells. Research that was actually, how to put it, borrowed without permission from a Jewish scientist by the name of Robert Remak, which led to two more feuding scientists. Thus, from teeth gunk to torquing off Newton, crystallization to Schwann cells, the cell theory came to be an important part of biology today. Some things we know about about science today may seem boring, but how we came to know them is incredibly fascinating. So if something bores you, dig deeper. It's probably got a really weird story behind it somewhere.
All right, I'll leave us on that little image there, okay? Before we go on, any questions about kind of the history of cell theory? It's all right if you don't have them. The one thing I'll talk about a little bit that they talk about that, if any of you end up working in a neuroscience laboratory, they brought up Schwann there. Has anyone ever heard of a Schwann cell? Yeah? Anyone want to say what a Schwann cell does? It's all right if you've heard of it. So those Schwann cells, yeah.
Neurons isn't it the thing that myelinates?
Very good, yeah. So you have like this little neuron. I'm going to draw like kind of a simplified version of it. And a neuron is a type of cell in your nervous system. And then it kind of, this is a very poor drawing of a neuron, but it generally kind of looks like this. But your nervous system needs to move messages super fast. And so what they have is these little guys around them called myelin. And that myelin helps the message go really quickly, but the things that make them are called Schwann cells. So he was involved with that, kind of that process of figuring out what cells were, but they named this after him. All right, so we're ready for our first quiz, everybody. Are you ready? This is supposed to be hopefully a little bit easy. Don't overthink it. On the thing that says Basic Biology Course, check for understanding. Does everyone have that? Okay, can you please do question 1? Try to do it just independently. So please do question 1. And then look up and give me a little bit of a thumbs up when you think you're done. All right. Yeah, the one that says Basic Biology Course, check for understanding. Just number 1, yeah. All right. So we're just going to self-grade this first one. Does anyone want to feel like they want to be brave and tell me what you think the answer to number 1 is? Yeah.
B, Anthony von Leeuwenhoek.
Yeah, exactly. So it's B. Antoine or Antony. There's actually a von Leeuwenhoek or Leeuwenhoek. You'll hear a lot of pronunciations, but he's widely credited with the discovery of the microscope. So if you got that correct, you could just give yourself a little check, or you can put a +1 if you want to, however you kind of want to record this. But if you got it wrong, don't feel bad. It's okay. You can put a little slash there. Maybe you thought it was Isaac Newton because his name got brought up there a lot. But I'm going to have you slowly start to record these, and then we're going to be able to put them all together to figure out which categories you're strongest in and which categories you want to work on the most, okay? So don't worry if you got it wrong, but we're going to move on now, okay? So next, I'm going to have you go to your big packet, and we're going to talk about the structure of cells, okay? We're going to do pages 1 to 4 in this packet, and I know it's this big, intimidating packet, but we're going to get through this. And actually, these diagrams, I don't know if you've ever used these POGL worksheets before, but I even use these when I had to review for some of my teacher tests, for some of my basic biology information. They're super, super helpful, and they help you to kind of retain the information. All right, any brave souls out there want to read the why for me? All right, thank you.
An efficiency apartment is a one-room apartment. This one room is where you sleep, eat, shower, and entertain your guests. It all happens in one room. It is a simple way of living in a small space. A mansion is a large, complex living space with many separate rooms. There are rooms for cooking, eating, sleeping, bathing, reading, watching TV, entertaining guests, exercising, and storage. The rooms in a mansion are constructed for the specific things you would like to be able to do. You can live in simple efficiency or complexity. In this activity, we will be looking at cells that are as simple as a one-room efficiency apartment or as complex as a mansion.
Thank you. Wonderful reading voice there. Good projection. All right, so question 1 here says, the 3 bacterial shapes in model 1 are referred to as coccus, or coccus, which is sphere, spirillum, and bacillus, which means rod. Label the diagrams in model 1 with the correct descriptions. So I'm going to pull this up, but we're going to do this all together. So if I pull this up, we're going to think about this. So the first one is coccus, which is sphere. Which one do you think is coccus or coccus? Yeah, which one do you think it is?
The first one is
Yeah, right? It's a sphere. So you can just put a little label to say coccus there. C-O-C-C-U-S. Has anyone ever heard of Staphylococci? Right? Staph infections, right? So that's actually a Staphylococcus, right? Cocci. It means it's a circular shaped bacteria. Okay, staph is a bacterial infection. All right, the next one is, let's do bacillus because it's labeled. Bacillus is rod. So which one is bacillus? Yeah, what do you think? Good, the rectangle, right? That's my rod-shaped one, okay? Bacillus is a very kind of common bacteria. You find it in a lot of soil bacterias. So that's going to be my bacillus. Many of you will work in bacterial labs, and you'll hear bacteria that have these really crazy names at the beginning, but they will often end with bacillus, or they'll end with coccus, or they'll end with spirillum. And it's actually just based on their shape. Okay? And you'll work in labs where you can often see these too if they have high-powered microscopes. So finally, what should we label this one? Spirillum. Good. All right, that's my curved shaped one. All right, number 2, what is represented by the small dots found in each of the bacterial cells? What do you think? Yeah. Ribosomes. Yeah. And what do ribosomes do?
manufacture protein.
Good, they make proteins. Hopefully you all know that. Number 3, what is the name of the outermost layer that forms a boundary around the outside of each cell? What do you think?
Peptidoglycan cell wall.
Yeah, it's the peptidoglycan cell wall. It's made with peptidoglycan. It's the cell wall. And that's just the thing that's kind of on the outside there. So remember, all bacteria have ribosomes. They all have a cell wall there. Number 4, how is the DNA described, and what does this mean? So look at the DNA in our little diagram there. How's it described?
It's described as free-floating, meaning it's not bounded by a nucleus, but rather it's a nucleoid, so it's just free-floating in there.
Exactly, right? Would you call these eukaryotic or prokaryotic cells? Yeah, because they don't have a nucleus, right? So they just have their DNA kind of just floating there. And it's not necessarily always DNA in a bacteria. Sometimes it's RNA, but they all have some genetic material because they're alive. And so we see the DNA, and they have their ribosomes. Do you see any other organelles in there? Do you see mitochondria? Do you see a Golgi apparatus? Do you see an endoplasmic reticulum? No, because these are prokaryotic cells. But they have to have ribosomes. Why do they have to have ribosomes? Yeah. Yeah, which I appreciate you saying that. They build everything. That's actually, I'm going to write it right here. That's the central dogma. We call that the central dogma. And it's a good idea just to know this. If you're in a lab, they'll say, don't you know the central dogma? And in high school, I didn't know the central dogma. But the central dogma is basically just DNA transcribes to RNA, very good. And that makes proteins, yeah. And we'll actually talk a little bit more specifically about this. But everything that you are is proteins. They say like DNA is the most important thing. It is, but only because it makes proteins. DNA doesn't do anything, right? It's just some instructions. The things that make the proteins are there. So this is why even the most basic cells often have to have ribosomes. Yeah? Yeah, it's kind of this idea that DNA makes proteins. They call that the central dogma. Because all living things either have DNA or RNA to make proteins. And there's one little exception there, though, right? Something that's just on the border of not living. Do you know what I'm talking about? Oh, RNA stands for, I'll actually write this down. We'll learn this a little bit more. It's ribonucleic acid. And DNA is? Yeah, deoxyribonucleic acid. We'll actually talk about those structures in a second. No, well, not in a second, probably after lunch. But many of you are going to work in a probably genetics laboratory. It's helpful to know the difference between those two. Okay? So in terms of living things, you either have to have DNA or RNA, some genetic material. But there's something that doesn't necessarily have DNA or RNA, but it's just almost not alive. Yeah, viruses. So the one kind of exception there is like viruses. And a lot of people say the viruses aren't living. Does anyone know why they say they aren't living? Yeah?
They aren't able to manufacture their own proteins or leptins so that they can sustain themselves.
Yeah, they can't do the central dogma, right? They can't make their own proteins. But what they do is they hijack cells, right? They go into cells and they get cells to make things for them. And we'll actually talk about that later on. But viruses are kind of like semi-alive. You're all definitely alive. You all have DNA that makes these proteins, right? Your whole body's made of proteins. Your eye color is the eye color it is because of the shape of the proteins. Your skin color is the color it is because of the shape of the proteins. Yeah.
They're not exactly an abiotic factor.
No, and the whole idea of like abiotic factor, biotic factor, it's almost too binary to even think about viruses. Yeah, but just think about it as they're like semi-alive. And most people, I tend to hear people argue that they're just not alive. They just are things that take advantage of alive things. But viruses are really cool. And if you ever work in a virus lab, there's a lot of different ways that viruses take advantage of things. Okay? All right. I digress. So let's go to the next page. Number 5, all the internal structures are suspended, floating in what substance? I bet you don't even need to look at that. What is it? Yeah, the cytoplasm. Or the cytosol. Is it labeled as cytosol? Okay. Or the cytosol, yeah, which is just the liquid there. All right, number 6, one of the bacteria in model 1 has a tail-like structure. Do you see that? Look at that guy. What is that structure called?
Flagellum.
Yeah, the flagellum. Yeah, it means that bacteria is flagellate. And what do you think the purpose of that flagellum is for? Yeah, to move. Other bacteria that don't have that actually sometimes can't move. They just kind of go with the flow. But these ones can, right? And then C, based on your answer to the previous question, what might you infer about the absence of this structure on the other 2 cells? Can they move? No, right? So how do I get, this is a foci, this is a circle. How does staph, I can't swim. How does it get into me if I get a staph infection? Yeah. Exactly. It's just kind of passed along, right? So that's not, it doesn't, I mean, most bacteria that we get they can't just swim into you. That's terrifying, right? They actually are just kind of spread through airborne pathogens. They're very light and they can just float around. Or they're on like things. Whenever I go to the gym, I'm always careful to cover up my arms because you can get a staph infection at the gym, right? If you touch the material. So these are all things that are just sitting there. If you have a little cut, it can get into your skin. You can get staph infection. Yeah. Yeah.
But how do we prevent the overgrowth from happening?
It's a great question. So you're right. Staph is everywhere. If I swabbed this table, I promise you I would grow some staph, right? And a lot of these bacteria are everywhere. It's partially about how many of them there are, because our body is able to recognize staph and say, that's not a part of my body, and it's going to kill it. But if our body misses it, or our body's unable to fight it off because we're immunocompromised, or we have too much of that staph, then that's where it's going to kind of defeat us. But in general, if you just get staph on your skin, I mean, all of us have Staph aureus on our skin. We're not going to get a staph infection from that because we don't have a cut on our skin. And even if we do have a cut, often our body fights it off. This is why we don't always have a staph infection. Yes, a staph infection is very, very common. Or strep, which is streptococcus, is also common. All right, let's jump to model 2. Question 7, look at model 2. Listed these 3 structural differences other than shape between the animal and the plant cell. So which one? Yeah, the presence of the chloroplast. Who has the chloroplast? Plant cell, because they do photosynthesis, which is another one.
The animal cells don't have cell wall.
Yeah, cell wall. Yeah, so the animal cells do not have cell wall. Animal cells can't. And what's the last one?
Contractile vacuole.
Yeah, so you see a, are you talking about this vacuole?
Oh yeah.
So it's actually not technically a contractile vacuole because a contractile vacuole wants to spit. I know that's good you're thinking of that. You might have learned about this in your living environment class. Contractile vacuoles want to pump out liquids. This vacuole actually likes to store liquids because plants need it for photosynthesis. But yeah, the large vacuole is a dead giveaway. But really, if you're looking at a microscope, it's the shape, right? Cells, I'm sure you've all looked at a microscope of a cell because you're in New York City. It's the shape. They come as a square shape, the plant cells, or a rectangle. All right, number 8, where do you find the DNA in each cell? In the nucleus. Good. So these are prokaryotic or eukaryotic? Eukaryotic. Yeah, because they have a DNA. And we'll actually talk about that in just one second. So number 9, do both cells in model 2 have a nucleus? Yes. All right. Sorry, I know I'm going very quickly. All right, number 10. List the structures that form the boundary between the inside and the outside of each cell in model 2. So what forms the boundary between the inside and the outside? Yeah. Yeah, the cell membrane. And I'll make a quick note. The cell wall in a plant cell does not form a boundary between the inside and the outside. The cell wall is actually very porous. Things can go inside and outside that very quickly. We know this because when a cell dries out, a plant cell dries out. So a normal plant cell, I'm sure you've all seen an onion cell. A normal plant cell, when it has plenty of water, will look like this. My green here is my cell wall, and the purple here is the cytoplasm. Okay? But when it dries out, it'll look like this. Exact same cell wall shape, but my cell membrane is there, is all kind of shriveled up. And the barrier of my cell is really here, not here. The cell wall is kind of just like a shell. It's not really what controls what goes in and out. And the cell wall doesn't have necessarily all these interesting things with diffusion and osmosis as the cell membrane does, or it doesn't have those hormone receptors, right? The cell wall it's very porous, right? It has little pores in it if you were to zoom in on it. All right, so number 11, what is different about the outermost boundary in plant cell compared to an animal cell? Plant cell has a cell wall. And 12, decide as a group whether the cells in model 1 or 2 are more complex. And I'm not going to have you list 3 supporting reasons. Which one's more complex, cell 1 or the model 1 or model 2? Model 2, right? Mostly because they just have more, what do we call these little guys? Organelles, right? You know why we call them organelles? Yeah, mini organs, exactly. Because we knew about the body's organs thousands of years ago, right? People in very ancient societies used to cut people open and they'd be like, that's an organ, right? That's a spleen, that's a heart. I know this is called an organ. And then when we started finding these out, we're like, well, those are little cute organs. So we're going to call them organelles, right? But in case you don't know that, we call those the organelles, right? All right, model 3, structural comparisons. This is where we get into our pro versus eu. Pro means before. Carry-on means nucleus or kernel, and U means true. A little bit of etymology for you. So 13, use the chart and model to 3 to determine the meaning of the word prokaryote. What does prokaryote mean? What does it mean, Frederici? Yeah, before a nucleus. Okay. And what does eukaryote mean? True nucleus. Okay. And 15, based on the above definition, label the cells in model 1 and model 2 as prokaryotic or eukaryotic. So in model 1, are these prokaryotic or eukaryotic? Prokaryotic, right? They do not have a nucleus. Their DNA is just kind of floating there. These guys have their DNA nice, comfy, cozy in their nucleus, so they are eukaryotic. All right. Number 16, by comparing model 1 and model 2, What structures are the same in both prokaryotic and eukaryotic cells? So we actually kind of went over this. Yeah. Yeah, they both have ribosomes. What else? They both have a cell membrane. They both have cytoplasm. Anything else? Yeah, what's one more? So they don't necessarily have nucleus, but they do have DNA. Yeah, so they have genetic material. And those are the big ones. To be honest, this worksheet is not good enough to do that, but it is true. You will find many cells who, it's just genetic material. It's not necessarily DNA. Okay? So they could have RNA, and they could actually make proteins just based on that. So some cells just don't have any DNA. It's easier at the high school level just to say everything has DNA, but some cells don't. And that's okay. They're still cells. All right. So then let's go to the back here. We're going to do this super quick because I'm going to help you. A bacterial cell, does it have a cell membrane? Yes. So we can put a check there. Does it have a ribosome? Yes. Does it have a cytoplasm? Yes. Does it have mitochondria? Do I see mitochondria here? The powerhouse of the cell? No. So keep that question in mind. I'm going to put it over here in our little cell energy, which we're going to talk about. Cell energy. Remind me I put it here. How do prokaryotes make energy? I bet some of you know this answer. And if you don't, it's good review. We'll come back to it. Yeah, I know some of you know it, but just keep in mind they don't have a mitochondria, but that makes energy. I don't know. Do bacterial cell have a nucleolus, which is like the centralized area of the DNA? No. Do they have a nucleus? No. Do they have DNA? Yeah. Do they have a cell wall? Yeah. Are they prokaryotic or eukaryotic? Prokaryotic. Good. And then I'm just going to give you a little hint here. The animal cell has all of it, right? Including the nucleolus. And the plant cell has all of it. But they're eukaryotic. So that means all cells do they all have a cell membrane? Yes, every cell has a cell membrane. If not, it would just be like a free-floating nothing, right? It's got to have something to keep it in. Do they all have ribosomes? Yes, because the central dogma says so. If you're living, you have to have ribosomes to make proteins. Do they all have cytoplasm? Yeah, you got to have some liquid inside. This is why we also think, one of the reasons why we think water is necessary for life. You got to have something filling up your cell. Mitochondria? No. Nucleolus? No. Nucleus? No. But DNA or some other genetic material? Yes. Okay? And and do all cells have cell walls? No, right? Because animal cells don't. So now you know what all cells have. All right? So we're not going to do 19 through 22, but what I'm going to have you do right now is do your best just to do, and you can use your notes, so this is actually probably going to be a little easy for you. I would like you to do where it's questions 2 and 3. You can use your notes, but you actually don't even need to tell me the function of the organelle. We will come back to that later. If you know it, you can fill it out. But if you could just answer questions 2 and 3. For question 2, just give me the name of the organelle. And for 3, what is the main difference between prokaryotic and eukaryotic cells? All right, take about 10 seconds to put your last answers if you need to. All right. What I'm going to have everyone do is I'm going to have you exchange with a partner. So everyone in this kind of row will change, exchange, and exchange. I don't do this to be mean. It's just sometimes good to have another eye on yours. And remember, you don't have to feel bad if you're getting some wrong. But we're going to grade these, okay? And then at the end, we'll put the total number we got right just for this page, okay? So, okay, ready? All right, so for 2A, 2A is the cell membrane. So if they got it wrong, you just put a little slash through A. If not, you can leave it alone. 2B is the cell wall, because it's on the outside of the plant cell. 2C is the cytoplasm. Although, did anyone put ribosome? Were you tempted to put? If anyone did put ribosome, if you put ribosome for that, I'll be lenient because there's those little dots around. If you put cytosol, you can get that too. All right. So for D, it's the vacuole. That one might have been tough for you, but just remember that both animal cells and plant cells have vacuoles, but it's really big in a plant cell because they need to hold the water for photosynthesis. E is my nucleus. And F is the hint there was it's only found in the plant cell. So that's my chloroplast. Okay? G will actually come back to. You don't have to grade G. But G is actually, does anyone know what that is? Yeah? Yeah, so those are the centrioles, or if you put like, if someone did put microtubule, because you're kind of thinking that way, that is also fine. All right, and then question 3 is the main difference between prokaryotic and eukaryotic cells is that eukaryotic cells have a nucleus, okay? Or if someone put the eukaryotic cells have membrane-bound organelles, that's also fine. But the big difference is that eukaryotic cells have a nucleus, okay? So you can, hopefully you were able to just put a little slash if someone got it wrong. But what I'm going to have everyone do is you can give the paper back, and we're going to count the number that we got right. And then we're just going to put it right at the bottom next to where it says 1. So if you got number 1 right, you get a point. If you got 2A right, you got a point. 2B, 2C, 2D, 2E, 2F, and then question 3. So that's a total of 8 points. So if you got everything right, you get 8 points. You don't need to grade G. And if you're not great on this, then you need to review types of cells, right? It's just going to be helpful for you. So don't feel bad. And it's all, I do want to know the grades at the end because it will just be helpful to tell the people who are running this. Just say, oh, you know what, they may need some more review on X, Y, Z. It's not going to be a reflection of your character at all, though, okay? All right, so we are ready to learn about photosynthesis. So if everyone can go back to the big packet, we're going to just talk about the first 2 pages of the photosynthesis packet, okay? And this is a very basic review of photosynthesis. But if any of you work in plant labs, you're definitely going to want to do some more research on photosynthesis, okay? All right, who wants to read my why? Who wants to read my why? Would you like to read it again? You have a wonderful projecting voice.
What would the world be like without leaves? No grass for both fields, no beautiful landscaping. It would also mean no oxygen for animals and no food for Eutrophiles.
Good.
Leaves are like living machines that recycle the carbon and oxygen in our environment. This process, driven by the sun's energy, allows for a constant supply of oxygen and food for the inhabitants of Earth.
Okay, very good. So this is the leaf sun catcher. And let's look at the 3 things that are entering the leaf in model 1. So number 1 is my sunlight, because the arrows are going in. Number 2 is carbon dioxide, or you could write CO2. And number 3 is, wait, I can't see it. Where is it? Yeah, it's the water coming from the stem. Okay. I have to tell my wife this all the time. When we're watering the plants, I have to because sometimes she likes to spray the plants, and I got to be like, no, you just got to get the roots because it's all coming from the stem, right? Even though it's satisfying to spray the leaves of a plant, you just got to go for the roots because it's all going to be coming in here. There are some exceptions to that, but I won't go too deeply into that. All right, number 2, list 3 substances leaving the cell. What are the 3 substances leaving the cell? I see oxygen. I see water, but this time it's as a gas. Okay. And then I see sugars. Yeah, the sugars are leaving out of the stem. Unfortunately for us, plants don't just release sugar into the air and we just absorb it, although that would be cool, but they make their sugars there. And they deliver it to the rest of the plant to either make new plants or repair themselves, or new leaves repair themselves. All right, number 3. Which substance is both entering and leaving? Water. Good. Veins are important structures that carry materials through the leaf. Label the central vein in the leaf diagram. So where's my central vein? That is?
Sunlight.
Yeah, the sunlight's kind of pointing to it. But yeah, if you could just label this central vein, where you see it kind of sprouts off there. Has anyone ever taken a botany class? Do you ever kind of talk about that? When you go to college, I recommend it, even if you're not super into plants, because you get to learn how to identify all these different plants. And it's amazing, because all these plants, based on the little shape of their veins or the shape of the outside of their leaves, you can actually identify plants very easily on that. It makes all these different cool combinations of plants. All right. So for the next part, how is the substance you identified in question 3, which was water, change between its entry and exit? Well, in its entry, it is liquid, yeah. And then in its exit, it is gas. Good. This is why when leaves are releasing their water, they don't just kind of drip water. Normally, if you see kind of water dripping on a plant, it's because it's condensing there, because there's a lot of water in the atmosphere. Okay? Number 6, use the general equation for photosynthesis in Model 1 to answer the following questions. So I have this general equation, which is carbon dioxide plus water. This light energy gives me sugar and oxygen. Okay? What are the reactants for photosynthesis? Good. CO2 and water and light energy. This means that it needs to take place with that. But light energy doesn't necessarily, it's not necessarily something you can like hold in your hand. So we often kind of see it put right there. And where do these reactants enter the leaf? Just good to know. Carbon dioxide is actually underneath the leaf. Yes, but this is the generalized form. Yeah, it's not different types of sugar. Yeah, well, actually, sometimes it can be, but in general, with photosynthesis, ultimately what you're making is glucose. But as you may know, it gets turned into all kinds of different complicated types of sugar based on the plant that it's growing. No, it gets transformed into the cell. Yeah. But then the other things that are entering into the cell are the sunlight, which comes from the top of a leaf. Okay. And then the water, which comes in from the vein or from the stem, ultimately from the roots. And the products of photosynthesis are sugar and oxygen. And those leave the cell. The oxygen leaves underneath the cell. And the sugars leave back through the vein. They don't list water, but actually in a more complicated version of the equation, you would see that. To be honest, every version of the photosynthesis equation, no matter how advanced your high school classes have been, are simplifications. It's actually like a crazy, crazy many step process. Even cellular respiration is like a way more than the process that even I will teach today. These are all generally simplified versions. And you may be working in a lab that if they're talking about cell energy, they may be talking about some part of those equations. But it's good to know the overview of it to try to figure out where it fits in. All right, yeah?
It's not only the central vein, it's all the other veins too?
Yeah, so these veins right here are going to go towards the central vein, which is going to go back to the stem. Yeah. But if you ever look at a leaf, often that central vein is the biggest one. But some leaves actually don't have that central vein. Some leaves, like a pumpkin leaf, are going to look like this. Or like lettuce, some lettuces. They don't necessarily have a central vein. They kind of shoot out differently. But they're all ultimately going to be going back to the stem. Okay? So for this next one, we have the cross section of the internal structure of a leaf. So for number 8, list the layers of the leaf starting at the upper cuticle all the way down to the lower cuticle. Okay? So we'll list these in order because this is just helpful to know. We have our cuticle. Then right underneath that, we have the upper epidermis. You've seen that word epidermis before.
Yes, it's like skin.
Yeah, you see it with skin too. And then you kind of can think about it as the skin of the leaf. Okay? And then we have something called the Palisade Mesophyll. I remember when I first heard that word in college, I was like, that's a big word. And I don't use that word very regularly, but it is helpful to know it's kind of the top layer. What are these little guys in the Palisade Mesophyll? Little chloroplasts, right? And that's actually why the sunlight comes from the top of the plant and not the bottom, because you don't see a lot of chloroplasts here. Okay? After that, we have the vein is here, but we can just put the spongy mesophyll. There's a lot of air space there, because the gas exchange, where carbon dioxide's coming in, oxygen's going out, is going to come from the bottom. Then we have the lower epidermis. And then we have the cuticle, or in this case, it would be the lower cuticle. Okay. The one other thing that I want to make a quick note about is the guard cell. I find that sometimes students don't learn about this. Some of you may have learned about it, but this is a very important thing for us to kind of understand in terms of why the leaf is structured the way it is. So we have this thing called a stoma there. And you'll notice that that's an opening. And that allows that gas to kind of go in or go out. Okay? And these guard cells are there that are just going to open and close it, open and close it. Okay? The gas needs to be exchanged. You need to have these openings because carbon dioxide needs to come in, oxygen needs to go out, right? That's a part of photosynthesis. But there's just something to consider. When you have an opening here, similar to like, have you ever fallen asleep with your mouth open? And when you wake up, does your mouth feel really dry? Because you released a lot of moisture from your mouth because you fell asleep with your mouth open. That actually is the same thing that happens with plants. If their stoma is open, they're going to have that gas exchange, which they have to do because this is airtight if not. But they're also going to lose a little bit of water. And so if you are a desert plants develop this really, really cool type of photosynthesis where they don't have this open very, very regularly. In fact, they actually only tend to open it at night when it is not so hot outside. So they collect all of their sunlight during the day, which is going to give them all the energy, which we'll talk about for photosynthesis. But this will actually remain closed, so they're not necessarily doing a lot of photosynthesis there. They're kind of just building up everything they need for it. And then at night, when it's cooler, so they're not going to be so dried out, then their guard cells open up, and then they have this gas exchange to kind of say, oh, now it's time to do our photosynthesis, where they can do that, and they lose less water. It's a pretty cool little fact for you. And if you're working in a photosynthesis lab, they may talk about, there's different types of photosynthesis. I won't go into them because we have a lot to cover, but that's just a helpful thing to know. Okay? All right. Before we go to the check for understanding, we're going to go to the next page, which, oh no, we're not. I'm just kidding. We're not going there. We are going to go to, I just want to make sure I didn't do this out of order. Okay. Our little photosynthesis thing is missing, which is okay. Let me just give you just kind of a super brief. Actually, you know what? This kind of introduces photosynthesis for what you need to know. So there's one little worksheet. It was just a one-pager that got removed from this, which is fine. I'm going to have you go back to the check for understanding. You can skip, because the information for number 4 was on that one. You can skip question 4 for now. And I'm going to have you do just question 5 and 6, okay? On page 2 of the Check for Understanding. While you do that, I'm going to pull up what I would need for the next part. D is pointing to, oh no, D is pointing, oh, I'm realizing that this didn't copy well. Let me, yeah, there should be a little line. Let me see. No, it's actually not that. Let me pull up that image for you. Well, I'll just, no, there, oh, this all got messed up. So you don't need a G. The D, I'm just going to give you, I'll draw it. It's a little bit of a hint. So in the cell, E is pointing to this, and D is pointing to what this would be. Hopefully that's helpful for the bottom of a cell. Ignore the G, sorry. I must have copied and pasted the first table. And this is the one page that didn't go. I actually just have this as like our top part there. It's pretty basic because you can kind of just copy it from there. But I'll just do the read this for you. This is not in your packet. This one page didn't copy. But inside the plant veins are 2 different types of tissue. The xylem carries the water and minerals up from the roots of the plant. And the phloem carries the sugars away from the leaf to areas where the plant is growing or to storage areas in the plant. The way that I remember that, I mean, if you take a basic bio class in college, they just will tell you about xylem and phloem. The way I remember that is, I don't have any way to remember the xylem, but I know that there's 2 things. And the phloem starts with a P. It carries the products of photosynthesis. It carries the sugar away. And the thing to remember is that the xylem is going to carry it up because it needs to happen in the leaves. And then once the products happen in the leaves, the phloem is going to return it back. So in a way, you can answer question 4 now. If any of you are done early, especially you AP biology students, page 3, I'm curious if you have that memorized yet. I don't know if nowadays you have to know all that stuff from page 3 for the, or just the number of everything. Page 3 of the Check for Understanding. Little hint of what's to come. All right, everybody. Can you please switch with your partner, and we're going to grade page 2. Okay? All right. Number 4. The big difference is the xylem carries the water to the leaf, or you could say carries materials to the leaf. The phloem carries it away from the leaf, or you could say the phloem carries the sugar. Okay? Hopefully everyone got that, because you could have just copied it. So the xylem carries the water to the leaf, or it carries materials to the leaf. The phloem carries it away from the leaf, or they could have said the phloem carries the sugar. Okay? So if they got that, you can give them a little +1 or whatever notation you want. If they got it wrong, give them a little slash so we can easily count it. All right. So for A, let's go back to our notes to see if we can get it. Okay? So for number 2, or question number 5 there, A was my cuticle, or they could have said upper cuticle if they're a real hero. Okay? B is, in this diagram, it's much smaller. So you kind of had to learn to think about this in a different sense. But B was actually my Palisade Mesophyll. Okay? C is much larger in this diagram, and that's my spongy mesophyll. You can know that because there's big gaps in that drawing, if you could see it. Those big air sacs. Okay? D is pointing to my guard cell. Hopefully that was not too confusing. And so E is pointing to the opening which is my stoma. And F then is pointing to my lower epidermis. Nothing for G. And 6, hopefully they said something along the lines of the guard cells open and close the stoma, right? That's what we're looking for. They open and close the stoma. You can't just always have that stoma open or you're going to lose your water. Mm-hmm. Yep. Yeah, so guard cells are going to respond to how much water is actually in the cells. And they will open if the cell is oversaturated with water. Yeah. But they're also going to respond to the relative concentrations of carbon dioxide or oxygen. If there's too much oxygen in the cell, that's waste for the plant cell, right? They need a little bit for their mitochondria, but it's going to respond to kind of help them out with that. All right, so if they got, let's count the number of total questions on this one, and they'll be able to write at the bottom. So for 4 is 1 point, 5A, 5B, 5C, 5D, 5E, 5F, and 6. So the total number is 8. So you can put that right down at the bottom by 2. And you can give it back to the person. And then that person can know if I didn't get a lot right or did not get 8 right here, I may want to review plant cells. Easy peasy? Yeah. I think so. Let me look that up. I'm not a good botanist for this. Yep. And this is what you should do if you don't know the definition of something too. Just Google it. All right, everyone, we're going to go back to the big packet. We're right on schedule. You're all doing great. Before I go on, and throughout this, if you have any questions, let me know. But any questions?
So the hypothermic, it happens when
You're thinking of, yes. So it's actually slightly different. So you're thinking of like when a cell is like hypotonic and it has too much. So I'll kind of go over that. That partially has to do with this, but not necessarily. That actually can happen with the amount of liquid that they take in. It's helpful to know these terms, so I'll just kind of go over them. Actually, I'll just do it down here. But if there's a normal amount of water around it, right, a plant cell, we would call this, same inside and outside. What? Isotonic, yeah. So this would be isotonic, meaning it's kind of same inside and outside. This is living environment stuff. Okay, but if a cell if there's much less water outside than there is inside, so let's say there's a lot less water outside, which means the water is going to want to leave the cell, right? Because there was a lot less water outside. So the cell has more water than the outside does. Do you know the term we use for this? It's actually kind of confusing. Well, the term is, yeah, the diffusion of water is osmosis. But in terms of the tonic waters, or tonic names, do you know? Yeah, it's actually hypertonic, which is a little confusing because you think of hyper because it has a lot more. And the truth is it did start with a lot more, but it shrivels up. So this would be hypertonic, which is more inside than outside. This is water. Okay? And then if there's more water outside than inside the cell, then it's going to flow inwards, right? And it's going to actually put some of that turgor pressure you're talking about, and my cell's going to kind of get fat. Luckily, our cells don't lyse like our body cells do, and it's going to get really full with water, right? Yeah, hypotonic, good. And that's more H2O outside, so it wants to flow in, okay? If this were my brain cell, Does my brain cell have a cell wall that can hold it in? No. So if I drink too much water, what happens to my brain cells? They explode. Yeah. And you could die. You could die from that. From what do we call it? Why am I blanking on this word? What do you call it when you're too thirsty? I'm dehydrated. Yeah. You would. I'm blanking on the phrase for when your brain explodes. But it's you're hyperhydrated. No, there's a different word for it. But when if you drink too much water, your brain can become oversaturated. This often happens with people who do drugs, and they are like drugs that make you thirsty. This is particularly common when you hear with like deaths from ecstasy. But ecstasy is a drug that makes you quite thirsty, and you don't feel satiated, so you just keep drinking water. And sometimes people can actually have so much water that their brain cells will start to lyse. Yeah.
So that's turgid or distended, and that's plasmolysed?
Yes, correct. Very good. Are you in AP Bio? Yeah. So those are helpful words to know for that. All right. So next, we're going to move on to cellular respiration. Okay. So that's going to bring me to cell energy. All right. So my why. Do you want to read again, or do you want to read? All right, perfect. We have you got an assistant.
Living organisms display the property of metabolism, which is general terms to describe the process carried out to acquire and use energy. We know that people need to eat, and in our food are various kinds of
Nutrients.
that our cells use. One large group of nutrients in our food is
Carbohydrates.
which supply our cell with glucose.
Which is C6H12O6. Yeah.
So the question is, how does the food we chew and swallow through our cells?
Okay, very nice. Thank you so much. So we're going to talk about how we get from the food that we eat to energy. Okay? And I know that this is taught a lot in a lot of courses, so this may be a review, but it's really, really helpful to understand this because we're soon going to talk about cell culturing, and it's just helpful to think about what cells need. And you may also be working in a lab that talks about cell energy. And you may have someone who's just studying NAD, right? Or who's just studying ADP, right? So these are things we're still finding out about. It's very exciting. We're going over the simplified things, but it's really helpful to know the overview so you can kind of zoom in on it. All right, so refer to Model 1. Did you refer to it? Great. All right, so what is represented by the hexagon? Glucose, our best friend, glucose. How many carbons are in one molecule of glucose? 6. Good. And glucose is C6H12O6. That's something you just got to know. Since freshman year for most of you probably. You just got to know. Maybe earlier for most of you. And that is right there in case you don't have it. So for 2, refer to Model 1. You refer to it? Great. Okay. A, what is represented by the triangle? Pyruvic acid. Now, if you are not in an advanced bio course, you maybe have never heard of that because we don't often talk about that for living environment. But by today, you will know what that is. So for 2b, how many carbon atoms are in one molecule of pyruvic acid? 3. And that's half as many as glucose. Huh, makes me think. Maybe there's a reason why it's half as many as glucose. All right, so for number 3, in the process of glycolysis, which is this process, what happens to the glucose? See my glucose there? After it crosses the cell membrane into the cytoplasm of the cell. So I have my glucose that just got in here, okay? And it can do this because the cell membrane is semi-permeable. It lets the glucose in, okay? But it gets let in, and as it crosses in, it turns into 2.
Pyruvic acid.
Yeah, and it turns into 2 of my pyruvic acids. And that's called glycolysis. And let me just tell you something that's very important about this. The glucose got let in. I'll go really quickly over kind of how this works. So I have my cell membrane, okay? And this is an oversimplification. It doesn't always work this way, but it's kind of a helpful way to think about it. And I have my glucose. And glucose is too big to get let into my cell. It can't just go in naturally. Does anyone know how it gets let in? There's a special hormone that helps it. Yeah. Yeah, insulin, right? When insulin, you need insulin if you are diabetic, yeah. And people who are diabetic are often are like, oh, my insulin level's too high, the insulin level's too low. But ultimately, they need insulin because they need to let glucose into their cells, okay? So what they do is they have these little receptors here. And if they have insulin, so insulin, which we also call, it's a hormone. And we also call this a ligand, or ligand, which is something that's going to kind of bind there. Once my insulin goes in there, okay, that's going to say, oh, this is the right shape, we're good. And then that will open up the gates here. That's going to say, all right, he's cool. The glucose comes in. If I don't have enough insulin in my body, or my body doesn't have enough, hasn't created enough insulin receptors, or some of those receptors have been killed off because I consume maybe too much sugar, which is type 2 diabetes. But type 1 would mean my body doesn't make enough insulin. I have to take insulin. And if I don't take insulin, what will not come into my body? Glucose. And we'll think about, just in a second, what happens if your body doesn't have glucose come into it. And then hopefully you'll be able to understand why the things that happen to diabetics happen to them. It's actually kind of simple to think about it, but we'll get there. So that's another question. What happens if no insulin? Okay? So that glucose gets let in here, and kind of through that process, we don't go too deeply into it, but it gets turned into 2 of my pyruvic acids, okay? Okay? So thinking through this, the only thing that I needed, the only kind of like organelle that I needed to do this was my cell membrane, okay? So now going back to our very first part, this was, I can just kind of show you. Going back here, can my bacterial cells do glycolysis? Can they do this? Do they have a cell membrane? Yes. Yeah, so they can, right? So how do prokaryotes make energy? They do just glycolysis, right? They can't do the other stuff that we're about to talk about, but they can do this. They can break down glucose into pyruvic acid, and we're going to talk in a second that releases a little bit of energy, and if you're a bacteria, you're super little, and that's all the energy that you need, okay? So this process, ooh, look at all these markers I have. This process is glycolysis. And to break that down, lysis is to break something. I'll make a quick connection to break. I'll come back here, don't worry. If this was my brain cell, and I had so much water come into it, it was hypotonic, so let's say this is my brain cell. And it was hypotonic. My brain cell will get filled up, not just my brain cell, but actually any cell in my body. It would get filled up, and then what would happen to it? It would explode. Do you know what that explosion's called? Yeah, it's called lysis. It would lyse. Yeah, because lysing, so it would explode. So it would just, it explodes. And we call that lysing. So lysis is breaking things, right? But it's not bad. It's not always bad. We need it. But lysing is to break it. So I'll say explode or break. Explode's a cooler word, though.
So does apoptosis go into
So apoptosis will lyse the cell, but it does it in different ways. So he's just asked about apoptosis, which is a process that our cells will undergo, particularly we learn about often with cancer, right? But it is planned lysis, right? It is saying, oh, you know what? I just got a mutation that's going to make me cancerous. I don't want to kill off the whole body, so I'm going to lyse this cell. Sometimes cells will self-lyse, and sometimes cells will, like a bigger cell will lyse them. But yeah, apoptosis is a type of it. All right, can I move on? All right, so going back here, glycolysis is the breaking of glyco, or like sugar. Yeah, glucose, exactly. All right. So going back here, let's go on to page 2 of the cellular respiration. Glycolysis occurs in the cytoplasm of cells and does not require the presence of oxygen. Therefore, the process is anaerobic. It is the first step used by cells to extract energy from glucose in the form of ATP. ATP can be directly used by cells. Anaerobic, you've probably heard this word before. If I'm at the gym and I'm doing anaerobic workouts, what kind of workouts am I doing? Weights, right? Because it doesn't require, a lot of heavy breathing, right? So I can just go, and then I'm not breathing a lot. The opposite of an anaerobic workout is an aerobic workout, which requires a lot of breathing. And what's an example of an aerobic workout? A lot of push-ups. Or running. Yeah, running's kind of the typical one. But anything that's going to kind of be like long distance, it's going to require you to breathe a lot, right? That's aerobic. So these cells also have aerobic or anaerobic things. But this first step is anaerobic. They don't need oxygen. And for this reason, a lot of bacteria that we have doesn't need to live near oxygen. With my freshmen every year, I always say, what are the requirements of all living things? And always many of the groups say oxygen. But do you need oxygen to live? No, you actually do not need oxygen to live. And there's some bacteria that live around no oxygen. And they just do glycolysis. If I go to Mars and I find some bacteria that live there, do you want to bet the way they get their energy is through glycolysis? Because there's not a lot of oxygen on Mars. Okay? Probably one of the reasons why if we find life on Mars, it's going to be a little bacteria that just use glycolysis, not something big that uses aerobic respiration, which we'll talk about. Okay, so for number 4, thinking about the number of carbon atoms in glucose, 6, and in pyruvic acid, 3. Explain why there is 1 molecule of glucose on the left side of the arrow and 2 molecules of pyruvic acid on the right side. So what happened to my glucose? It got split into 2. It got split in half. Kind of. I asked them if they wanted me to teach a lot of biochemistry, which would have been all day, and they said no. But if you want to really know kind of what happens, that would be a little bit of biochemistry. And I'm assuming all of you, some part of you wants to be a doctor someday. And if you want to be a doctor, you will have to take biochemistry in college. And it's great. It's interesting. And biochemistry is really helpful. I wish they taught it before biology, because it makes biology easier to understand. Okay. So number 5, how many ATP molecules are produced during glycolysis? So I started with ADP, which is something called adenosine diphosphate. And then that turns into ATP, which is the higher energy form of that. So I made 2 ATP molecules, which in the scope of living things is not a lot of ATP. You can't do a lot with it. But if you're just if you don't even have a flagella, you don't need a lot of ATP. The only ATP you really need is to make proteins, right? You're just going to sit there and you're going to make your proteins. But you don't necessarily even need that ATP. But if you're an organism like us, 2 is not going to cut it. So you need to do something more with that. All right, number 6. Hydrogen-carrying molecules are also produced during glycolysis. Okay? Hydrogen, the molecular letter for that is H. So something picked up a hydrogen. What was it? Yeah. So NAD got turned into NADH. So the symbol of the hydrogen carrying molecules, you could say it's a diamond here, or you could say it's NADH. All right. And those are actually kind of important. If all you learned about this was freshman bio, I'm assuming you probably didn't learn about that very much, but those are actually quite important when you figure out this process. So number 7, does glycolysis incur inside or outside the mitochondria? Is there a mitochondria? There's mitochondria here, but does glycolysis occur inside of it? No. So you don't need a mitochondria for glycolysis. Okay? Which is why these can do it. Can these, my plant and animal cell, do glycolysis? Yeah, they can because they also have a cell membrane. And they do glycolysis. All of your bodies right now are doing glycolysis. Okay. So, model 2, my Krebs cycle. Some of you have heard of this. Some of you have not. If you're like me, you're all junior. Are you juniors, seniors? Juniors. Is everyone a junior here? Some seniors? Yeah. If you're like me, actually, no matter what, junior or senior, I had never learned about the Krebs cycle until college. But it's cool. All right. And I wish I had these diagrams with me when I learned them in college. It's hard to learn from a textbook. I find these diagrams more helpful. All right, number 8. According to model 2, what happens to pyruvic acid during the Krebs cycle? So remember, my pyruvic acid is this. So it moved into the matrix, and then it gets turned into, it gets turned into carbon dioxide. According to model 2, where does the change identified in the previous question occur? It occurs in the mitochondrial matrix, the matrix. Not as cool as the movie, but still pretty cool. Number 10. Note the number of atoms of carbon in pyruvic acid and explain why 3 molecules of carbon dioxide are produced. Carbon dioxide, molecular formula is CO2. So how many molecules of carbon dioxide of carbon does carbon dioxide have? 1. And pyruvic acid has 3. So pyruvic acid can make 3 carbon dioxides. Okay? All right. Number 11, there's a key next to it. It must be important. Considering that glycolysis produces 2 pyruvic acid molecules per glucose molecule, I think we should write this down. All right, so glycolysis made my 2 pyruvic acids. All right. How many total CO2 molecules will be produced from the complete breakdown of each glucose molecule? So each pyruvic acid will make how many carbon dioxides? 3. So this will make a CO2. I'm going to make another one. I'm going to make another one. I'm going to make 3 here. So I will make a total of 6 CO2. And if any of you have the kind of the AP Bio or Advanced Bio formula for photosynthesis memorized, you may recognize that it's not just 1 CO2 that's made. It's more than 1. And this is part of the reason why. Yeah? All right. So for 12, what 2 hydrogen-carrying molecules, remember we're just looking for the letter H, are formed during the Krebs cycle? So what has an H that didn't have an H before? Yeah? Yeah, exactly. So NAD plus got turned into NADH, so it took on some hydrogen. And FAD got turned into FADH2. So you could say the energy-carrying molecules that were created were NADH and FADH2. You don't need to know what those do yet, but it's helpful to know that when you bring on a hydrogen, you're actually bringing on a little bit of energy. And you're going to be able to use that energy later on. So what this process does is while I'm breaking down my pyruvic acids, I'm using that energy to make these higher energy things. Just think about that H as the little pocket of energy that something brings on. Okay? We also see this in photosynthesis, although one thing that you often see kind of carried on to things in addition to these H is my P, my phosphate group. So if you're carrying a P or you're carrying an H, it's like a little pocket of energy that you can use for things. Guess what we're going to use to make ATP? All right. So let's do the math here. Okay. So for question 13, fill out the chart by looking back at the entire process of glycolysis in the Krebs cycle to list the total number of ATPs and hydrogen-carrying molecules produced. Okay? This seems like a hard question, but it's not. So in glycolysis, how many ATP molecules did I produce? 2. So you can put a 2. In glycolysis, how many NADHs did I make? 2. So in breaking down my glucose, I made energy, right? And when I grade the living environment test every year, we always ask, what's energy? And some students, if they're confused, they say glucose is energy. Glucose isn't energy, but it's used to make energy. Also on that test, we always say, what's the high energy? What's the energy of the cell? And the answer that everyone gives is, you know, if I said, what's cell energy? Yeah, you would say ATP. Everyone says ATP. And then there's always a couple brainiacs that say NADH. And I'm like, all right, give that person 100% on that test. But next is FADH2. Did I make any FADH2 in my glycolysis? No, so you can put a big 0 for that. All right, the next row is the Krebs cycle. Looking at this, let's figure out how many ATP I made. How many ATP did I make? 1. So this is just the first cycle. But guess what? In the second cycle, it's the exact same. So you can put a 1 and a 1. How many NADH did I make? 4. Ooh, this was a lot more energy. And I made 4 and 4 for the second pyruvic acid. Okay. And next, how many FADHs did I make? 1. I made 1 and then another 1. Okay? So breaking down that glucose is releasing a lot of things. So glucose is a very high energy molecule, but it's not actually the energy that's useful to us unless we put it into NADH or FADH2. Our cells can't just take glucose and use that energy directly. We need these other molecules to drive us. All right, let's go to the next part, Model 3. The electron transport chain. All right, so this is a little diagram here. This is the next step of cellular respiration. We have our outer mitochondrial membrane there. We have our mitochondrial matrix. We have a couple of things there. So let's look at this. What cell structure is the site for the electron transport chain? So the electron transport chain is this electron kind of moving through here. What is the site for it? We could say it's kind of the mitochondrial matrix. Now it occurs within the mitochondrial membrane, but it kind of occurs throughout this whole mitochondrial matrix. Okay? Number 15, label the carrier proteins in Model 3. So, thinking about this, we have, sorry, I'm blinking. Let me come back to 15. Number 16, which substance do the carrier proteins transport across? Oh, sorry, the carrier proteins are these. Okay? So you can just label those as my carrier proteins. Which substance do the carrier proteins transport across the inner mitochondrial membrane? Yeah, they're moving my Yeah, hydrogen. And particularly, it's the hydrogen ion. It's H+. And that's a very charged molecule. And they're using that charge to drive something. Okay? So they're carrying my hydrogen. Here they're pumping it out, and here they're actually pumping it in. All right. Read this. NADH and FADH2 molecules release hydrogen ions that are transported across the inner mitochondrial membrane with the help of electrons. So you see my NADH got turned into NAD plus here. It lost its hydrogen, and that hydrogen got released here and shot out there like a little mail tube, like, and it got shot out there. Okay? The result of these multiple processes is the production of a large amounts of ATP. So ultimately, I'm making more ATP here. They kind of do this chain where they're getting rid of their hydrogen, and then that hydrogen moves back here, and it wants to come back in, and the release of that potential energy is going to give me enough energy to make my ATP. How it actually works is it actually gets this guy spinning. As you move down it, it actually starts to twist that, and the energy from that twisting is going to help to power this. It's a pretty cool system. And like I said, this is just kind of a basic overview, and you'll have this to take this with you. And if you work in a lab that talks about mitochondria, you're definitely going to want to review this, because they're going to expect that you know about the electron transport chain, et cetera, et cetera. You don't have to dream about this, but it is helpful to know it. Okay? So number 17, what high energy molecule are formed by the electron transport chain? So just as a quick review, is NAD plus a high energy molecule? Is it higher energy than NADH? No, it's actually lower energy, so that's not high energy. Is FAD high energy? No, it's a lower form of FADH2. Is ATP high energy? Yes. ATP is the high energy. And ATP is the gold mine, right? That's what we want to power our cells. So for 18, refer to Model 3. What atom accepts the hydrogen ion at the end of the electron transport chain? What accepts it? Yeah, oxygen's going to collect it. And what gets formed as a result of that? Well, if I add 2 hydrogens to 1 oxygen, I get H2O. What's H2O? Dihydrogen monoxide. What's that? Water. You know that. Yeah, it's water. Okay, so I make water. Ooh, voila. All of you kids who have to have this memorized, you now know why cellular respiration makes water. It's basically just kind of taking these back. All right? And this water is not charged because this may be a plus, but that electron that is kind of directing this is actually going to be holding that as well too. So overall, this is going to kind of be a neutral substance. For 19, formulate an explanation for why the events of the electron transport chain constitute an aerobic process rather than an anaerobic process like glycolysis. One more time. If I am doing an aerobic exercise, I am using, I'm breathing in oxygen. So if something's aerobic, I am breathing oxygen. So if an aerobic thing requires oxygen, does this require oxygen? Right at the very end, I need oxygen. So this is an aerobic thing, okay? So if you are an organism that wants to do the electron transport chain, you need to do this. If you're an organism that is like, you know what, I'm satisfied with my glycolysis, I'm just going to get my 2 little ATPs from there, I'm a pretty easygoing organism, then you just need that. But if you are going to be a high energy thing, making a lot more ATP, well, then you're going to need to be in the presence of oxygen for this process. We do this, right? And this is why we need to breathe. When I'm exercising a lot, I need a lot of energy, okay? And that energy is in the form of ATP. So if I'm running, I'm going to use up a lot of ATP. And that's why after a long time of running, I need to make more ATP to keep myself going. And to make more ATP, I'm going to need that oxygen to keep this cycle going. This is why you breathe a lot when you exercise, when you do aerobic exercises. Okay? All right. I'm very impressed. We're going through this super quickly. This would take many people a week, and we're just going through it in about an hour. OK, so the next part of page 5, read this. I'll read it, just because I'm going to break for just some explanations. So remember that glycolysis produces 2 pyruvic acid molecules per glucose, along with 2 of the hydrogen-carrying NADH molecules. I'll go back here. OK, so I made 2. Sorry, that's not glycolysis. I made 2 of my NADH. Remember also that the Krebs cycle produces NADH as well as another hydrogen carrier called FADH2. So I keep on my first 2 steps. I'm just making a bunch of this NADH or FADH2. It is important to know that during the electron transport chain, when each NADH gives up electrons in hydrogen ions, okay? So here, the thing that's kind of driving this is my hydrogen that I'm losing from these things. So I build those up, and I just lose them at the end. There is enough potential energy change to make 3 ATP molecules. So this should actually say times 3 there. If you can all go back a previous page and just write down times 3. So every NADH can make 3 of those. There's enough potential energy change to make 3 ATP molecules. When each FADH2 gives up electrons and hydrogen ions, there's enough of a potential energy change to make 2 ATP molecules. So can you actually go back and where you wrote times 3, put a little dash and put NADH2, or sorry, NADH, because NADH can do that 3 times. And put a little times 2 as well and just make a note that that's FADH2. All right. We're going to do some math to figure out how many ATP molecules you can make with 1 glucose. Okay? So we're just going to talk about 1 glucose. So in glycolysis, how many ATP molecules are made? For my first box. In glycolysis, I made 2 ATPs. So for right there, you can put 2. And then let's talk about in glycolysis, how many NADHs did I make? 2. And how many FADHs did I make? 0. All right. Next, in the Krebs cycle, how many ATPs did I make? 1. And, oh no, sorry. It's not just 1. It is 2. Because that's for every pyruvic acid. I make 1, right? So it's 2. We're just seeing if you're paying attention. And, sorry, did I say FADH or NADH? Oh, sorry, did I say ATP just now? OK, sorry. Yeah. ATP, you make 2 of those for 1 molecule of glucose. And now for NADH, for 1 pyruvic acid, I make 4. For 2 pyruvic acids, I would make 8. Okay. For FADH2, for 1 pyruvic acid, I make 1. So for 2, I make 2. All right. Let's do the math here. So in glycolysis, how many ATPs did I make? 2. In the Krebs cycle?
2.
I made 2. OK. And now, how many NADHs did I make? Sorry, this is going to give me a total of 4. NADHs, I made 2 in glycolysis. And then for the Krebs cycle, how many did I make? 8. OK. Now that's going to give me a total of 10. Okay. But what am I going to do with that number 10?
Multiply.
Multiply by 3, right? Because every one of those can actually give me enough energy here to make 3 ATPs. Okay? So I'm going to multiply this by 3, and that's going to give me 30. So already I know that this process can give me 34 ATPs. Whoa, that's a lot more. Think about glycolysis only gave me 2 ATPs, right? But if I'm using oxygen and I have a mitochondria, think about how much more ATP I can make, how much more energy I can use. Now for FADH2, just FYI, I do not have these numbers memorized, so you don't have to memorize them either necessarily. But for glycolysis, how many did I make? 0. How many did I make in the Krebs cycle? 2. And I'm going to take that number 2 and multiply it by 2, which is 4. Good. So in total, I'm going to make 38 ATP molecules throughout this whole process. Okay? I make 38 ATP molecules compared to these guys who can only have the ability to make 2 for every glucose molecule they can. That's very inefficient. If you have a mitochondria, think about how much more efficient that is. Okay? All right, look at the equation for cellular respiration, and right in each stage of the process, each molecule is either used or produced. So glucose, let's do this super quick. My first one, C6H12O6. Do I use it in glycolysis? Sorry, I use it in glycolysis, right? Do I get to use my glucose anywhere else? Nope, because it's already been broken down. Okay? Exactly, yeah. Now oxygen. Is there any oxygen here? Do I see that little dot? No, it's not in glycolysis. Do I see that little dot in Krebs cycle? I don't see any oxygen there. Wait, do I use any oxygen here? No, yes, I do, right? So my oxygen is used in the electron transport chain. You could put ETC if you would like. And next, let's talk about carbon dioxide. Okay? Is carbon dioxide produced here? No, I don't see any carbon dioxide there. Is it produced here? Yep, that's right there. So it's produced in the Krebs cycle. All right. Next, water. Let's start from the top. Did I make any water here? No. Did I make any water here? No. Did I make any water here? Yeah, right at the end there. So I make water, I produce water in the ETC. Now ATP. I bet you're getting exhausted of this. Okay, for this, did I make ATP here? Yes, so glycolysis, I make ATP. Did I make any ATP in the Krebs cycle? Yep, so you could put the Krebs cycle. And did I make any ATP in the electron transport chain? Yeah, I made a lot of ATP there, right? That's where a lot of it comes in. Sorry, I should put ETC here. Okay, so compare the ATP available to cells when oxygen is present versus when it's absent. If oxygen is absent, I can only do glycolysis. If oxygen is absent, I can only make how many ATPs? 2. If oxygen is present, I can make 38. Ah, this is why so many things use oxygen. Okay, so if you were suffocated, or on a nicer scale, if you were working out so hard that you couldn't get enough oxygen to yourself, what would happen? So actually, suffocate is kind of the more helpful thing. Let's think in terms of our brain. If I am suffocated, I am not going to have oxygen. So I'm only going to make about 2 ATPs for every glucose molecule I have. Is that enough, do you think, to sustain your body, just in terms of the ratios? That's only making 2, and normally I need 38, which is just enough to keep me alive. So if I'm suffocating, I cannot make enough glucose. So if I'm suffocating, I cannot make enough ATP. If I'm suffocating, I can't do things in my body. Everything in your body is run by ATP. If it takes energy, it takes ATP. So if you suffocate, what will happen to your cells? They die. Yeah, they don't have the, they can't do, well, really, ultimately, they can't do the central dogma, right? They can't make things. They can't make things happen. So if you suffocate, you die, right? If you even suffocate for a little bit, right, you can have some cell death.
And then it affects the metabolism?
So ultimately, yeah. The problem is if you have a lack of oxygen, you can't metabolize glucose efficiently. You can't get all of the ATP out of it. So your body may try to break down that glucose, but it's not going to be able to do particularly that electron transport chain. So you can't get that 38. You'll still, until you die, you're still actually going to bring in those glucose and try to do it, but you won't. You'll eventually just die, right? So I'm going to bring all the way back to our first question, okay? If I don't have insulin, will the glucose be able to enter into the cell? No, right? It doesn't know the person that's running the door, and it's going to be locked out there. And if I don't have insulin, will I be able to create any ATP? No. And what happens if you don't create ATP? You have cell death. So what happens with people who have diabetes? What dies on them? Not necessarily just them, but what will happen with someone who has diabetes? Yeah. Yeah, often they need like a leg amputation. Very good. And they have like a lack of circulation. So what will happen is they don't have the necessary energy to keep all of those cells alive. So you'll see a lot of cells, like their feet or sometimes their fingers, will die and they'll need to be amputated because they're not getting enough ATP. You need that ATP to survive. Okay? So if you're diabetic, even type 1 or type 2, you may be eating plenty of glucose. But if you don't have that insulin, you can't bring it in. And then bad things can happen. You can go into a coma. You can have brain death. You can lose a limb because you're not having that. Okay? No matter how much glucose you eat, it's not going to help you. Okay. So, last page. Congratulations. We're going to do this one more page, and then we're going to go to the check for understanding, and then we're going to do lunch. So, you've made it. I think this is the hardest stuff. All right, so this is the cool part. This is where we want to get to, because this is how you make beer. This is how you make alcohol, right? This is the interesting thing. This is also how you make yogurt. But this is anaerobic respiration, particularly fermentation. Also how you make sauerkraut, how you make kimchi, how you make all kinds of pickled things, fermentation. Okay? So what are the two substances that may be formed in anaerobic respiration? I can either make lactic acid, or I can make alcohol and CO2. Okay? Notice here, glucose, pyruvic acid, all good there, right? We know that that can happen without oxygen. And then the ultimate thing that'll get created if you don't have oxygen, it doesn't just stop there. It'll do one more thing. It'll either make lactic acid or it'll make alcohol and CO2. Alcohol and CO2. That is so interesting, right? There's billions of dollars that are made based on that industry, including something that's not alcoholic. Do you know anything that ferments that we use the CO2 for? It's really helpful. Yeah? Excellent. Yeah, to make bread. The yeast that is doing this, the yeast is not doing aerobic respiration. It's just doing anaerobic respiration. But that CO2 the yeast makes my bread nice and yummy, right? It's going to make it all fluffy and it's going to release those air pockets. So when you cut open bread, it's kind of spongy, right? That's the CO2 that was made. This yeast actually makes a little bit of alcohol too, gets burned off. But sometimes if I put yeast in a container and I seal that container, it's going to release CO2 and it's going to release alcohol, and I've made champagne, right? I've made bubbling, something that is bubbling with big bubbling because the carbon, it's going to be carbonated, plus it's going to have alcohol. You're all too young to consume this, but that is how the industry works. And honestly, you all may end up in a lab that is interested in alcoholic fermentation. And that's a very important thing, particularly in our culture, because a lot of people in the world, a lot of people make a lot of money off of making alcohol. Okay? All right, number 24, recall what 2 molecules of ATP are formed during glycolysis. Oh, sorry, recall that 2 molecules of ATP are formed. Neither fermentation process shown above creates any more ATP. So, I made 2 molecules of ATP here, and then it made this one more step. I already made the ATP I needed. That's the purpose of all this. So why do you think they needed to do one more step? Why do they need to do one more step? If they got all the ATP they needed there, why didn't they just stop at pyruvic acid and just leave the pyruvic acid sitting there? Why did they need to turn it into lactic acid or alcohol and CO2? Yeah. To what? Energy that is going to be, actually no, not necessarily. It actually kind of absorbs energy. Yeah. So it is partially the waste, but actually the pyruvic acid is my waste. But let's go back here, okay? Let's look here. And this is a cool thing, and this is actually super helpful for especially anyone taking AP, it's helpful to think about it. When I did this, okay, I needed, my ADP got turned into ATP. I'm happy. And that ATP is going to go do a bunch of stuff and then get turned back into ADP when it has used its energy. But I produced something else. I produced NADH. And that NADH was really important for me in order to do this process. Now, if I only do glycolysis, do these NADHs have anywhere to go? Do they have an electron transport chain to release their hydrogens? They don't. They don't have an electron transport chain. And I need those NADHs to do it. So these NADHs need to somehow get back to form NAD. Where do you think that H goes? These need to get back there. And so they actually need to become lower energy. Where do you think they drop that H off? Not the glucose. And what? Yeah, they drop it off. They kind of give it to the lactic acid. They give their energy to the alcohol or the CO2, right? It's actually giving away the high energy molecule so that they can redo that cycle. That NADH is not super helpful to them. They actually need to get rid of that energy so that they can continue on this cycle, okay? So they need to create NAD again. They need to get rid of the H that's attached to them. All right. So quickly for number 25, just an interesting question. Research, we're just going to say, when you are overexerting your muscles, okay? Like if you're doing a bunch of crunches, right? Or you're running or you're sprinting and you can't breathe fast enough in order to get enough oxygen. Has anyone ever felt the burn when you are working out, right? It feels kind of burning. Where do you think that burning comes from? Which of these things would maybe seem like something that's burning? What? So not necessarily the alcohol, because I don't get drunk when I work out. I thought that too. No, and actually that makes sense, because maybe it would be. But if that were the case, if our body did this one, luckily it doesn't, then I would sprint and I would get drunk, right? And then we don't want that to happen. But where does it come to? Lactic acid, yeah. So that burning sensation actually can come from the buildup of lactic acid. And that's also something that can help to make you sore. So if you've worked out very hard the day before, that lactic acid can kind of build up and it can cause that soreness, which is a little bit of like a long-term burning. Yeah?
Doesn't that happen in some people? Like if they have this disease and like it just builds up and they become drunk all the time?
Yeah, so what you're talking about, it actually tends to happen with people who are like home brewers. So people who consume a lot of yeast. What they do is actually, our cells actually don't do this. They don't do alcoholic fermentation. That tends to be like yeast. But so our cells do this. If they don't have enough oxygen, they do lactic acid fermentation. But if I actually have a lot of yeast in my body, this is not like a yeast infection. That's different. But if I have a lot of yeast because I consume it, I'm going to have a bunch of yeast that actually could potentially be doing a lot of alcoholic fermentation in my body. And yeah, this is a super rare condition you're talking about, but it is a condition where people will get drunk just every time they like eat bread, because it's just going to get broken down, and then the yeast in their body does this. But that's like people who are consuming an extremely abnormal amount of like raw yeast, which particularly is like people who make their own, maybe like cider or things that involve yeast at their home, and they're eating it in a pretty raw state. It's not bakers, because the yeast dies in the baking process. Okay, so 26. What common foods involve the process of fermentation? Well, I kind of listed some of them, but what are they?
Bread, because yeast produces CO2, which causes the dough to expand.
Yeah, bread, any kind of yeast or any kind of dough that rises is going to involve that. What else? Alcohol. Yeah. Pickles. Yeah, that's fermentation. Anything else? Lactic acid. There's some things that make lactic acid, and it makes kind of gives it a it can give milk a sour taste. Do you know what that would be? What's like sour milk? Something that we would eat with sour milk. What is it? Yogurt. Yeah. So yogurt is going to involve this process. Kimchi is going to involve this process. It is really, really common in a lot of things. And luckily for us, it's delicious, right? We like the taste of that. And many people like alcohol, right? It is, alcohol is widely consumed around the world, and it's because we know about this process. But you need to have things that are going to be using these processes, okay? Cheese, yes, cheese does do it, but cheese also is slightly different. There's a bunch of different bacteria that are going to be using these processes. But yes, cheese is also going to be using lactic acid fermentation, or the bacteria that help to make cheese. All right, give yourself a deep breath. You've made it. You know everything about cellular respiration here. What I'm going to have you do next is page 3 of the Check for Understanding.
So for this question, can I also say that it mitigates the presence of like free radicals too?
Yeah, that is helpful.
But it's like a tribute, so should I remove that?
That is fine. Oh, I'm sorry. 13 is incorrect. Yeah. For the question for 13 should not say if the electron transport chain does not produce ATP molecules. Sorry, it should say if fermentation does not produce ATP molecules. Sorry, I was. So yeah, just one more time. 13 should say if fermentation does not produce ATP molecules, why is it used in cellular respiration? Oh, 7 gave you a little star there. My apologies. That gave you a star on the right answer there. That printed. That did not happen on the printer. It must print the correct answer. So you get a little gimme with 7. And just as a reminder, questions 10, 11, and 12 are per glucose molecule. So not in one round of the Krebs cycle, but per glucose molecule. Okay, take about 1 more minute. Almost done, like 2 minutes. Yeah. Okay, put your last thoughts down. And let's switch, okay? Switch. Number 7, that's my bad that that printed incorrectly. But the energy found in ATP molecules directly came from organic molecules, which means they're carbon-carrying molecules. So 7 was B. That's why there's a star next to it. 8, would an organism without a mitochondria be able to complete glycolysis? Yes. Number 9, would an organism without a mitochondria be able to complete the Krebs cycle? No. Number 10, how many molecules of ATP are produced with glycolysis? With glycolysis, I make 2. See? Right there. And how many molecules of ATP are produced with the Krebs cycle? You might have wanted to put 1, but the real answer is 2. Because this is per pyruvate. All right, number 12. Overall, how many molecules of ATP are produced from 1 glucose molecule? 38. Okay. And 13, ultimately, it's to reproduce NAD. It's to make more NAD. Or it could be, you could say to make more hydrogen-carrying molecules, but it's to go back and give you more NAD for number 13. So we'll count out the total number. Count if they got 7 right, 8, 9, 10, 11, 12, and 13. So the total number is 7. And if you could put that down by the page number 3 there. All right, we've made it to lunch. And when we get back from lunch, we're going to talk.